Hello everyone, myself Kirti. I am a student of BSc Chemistry Honours final year. This is the audio recorded lecture on the topic fermentation. Fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts, which are produced by microorganisms such as molds, yeasts, or bacteria. Fermentation technology is the use of microorganisms to produce food, pharmaceuticals, and alcoholic beverages on a large scale industrial basis. The science of fermentation is called zymology. Next is principle of fermentation processes. The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by providing raw material, meeting all the necessary requirements such as carbon, nitrogen, salts, trace elements, and vitamins. The end product formed as a result of their metabolism during their lifespan are result into the media which are extracted for use by human being and that have a high commercial value. Next is types based on respiration. First is aerobic fermentation. Aerobic fermentation or aerobic glycolysis is a metabolic process by which cells metabolize sugars via fermentation in the presence of oxygen and occurs through the repression of normal respiratory metabolism. Aerobic fermentation means that oxygen is present. Wine, beer, and acetic acid, vinegar need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much oxygen as possible creates a healthy, flavorful vinegar with the correct pH. Next is lactic acid fermentation. Lactic acid is formed from pyruvate produced in glycolysis. NAD positive is generated from NADH. Enzyme lactate dehydrogenase catalyzes this reaction. Lactobacillus bacteria prepare curd from milk by this type of fermentation. During intense exercise, when oxygen supply is inadequate, muscles derive energy by producing lactic acid, which get accumulated in the cell, causing fatigue. This is the reaction which show the conversion of glucose into 2-pyruvate and then to lactate. Next type of fermentation is alcoholic fermentation. This is used in the industrial production of wine, beer, biofuel, etc. The end product is alcohol and CO2. Pyruvic acid breaks down into acetaldehyde and CO2 is released. In the next step, ethanol is formed from acetaldehyde. NAD positive is also formed from NADH, which is reused in glycolysis. Yeast and some bacteria carry out this type of fermentation. Enzyme pyruvic acid, decarboxylase, and alcohol dehydrogenase catalyze these reactions. This is the reaction which show the conversion of glucose into 2-pyruvate and the conversion of 2-pyruvate into acetaldehyde and then 2-acetaldehyde into ethanol. Next is citric acid. Citric acid or 2-hydroxypropane 1,2,3-tricarboxylic acid derives its name from the Latin word citrus, which is a molecular weight of 210.14 grams per mole, which contain 3 carboxylic functional group with 3 different values of pKa, that is 3.1, 4.7 and 6.4. Citric acid is a primary metabolic product, is the most important organic acid produced in large quantity and is extensively used in food and pharmaceutical industries. Citric acid is tricarboxylic acid formed in the tricarboxylic acid cycle or Krebs cycle and is found in small quantities in all living organisms. Citric acid was first commercially produced in England in 1826 from lemons, which contain 7 to 9% citric acid, until 1990. It is produced mainly by submerged fermentation using Aspergillus niger or Candida from different sources of carbohydrates such as molasses and starch based media. This is the structure of citric acid. Structure of citric acid contain 1 aldehyde group and 3 carboxylic group. Next is citric acid production. About 99% of world citric acid production occurs through microbial processes, which can be carried out using submerged or surface cultures. The 70% of anhydrous or monohydrate form of citric acid is used in food and beverage industry as an acidifier or antioxidant to preserve or enhance the flavors and aromas of fruit juices, ice cream, and marmalades. 20% is used in the pharmaceutical industries as antioxidant to preserve vitamins, effervescent, pH corrector, blood preservative, or in the form of iron citrate as a source of iron for body as well as in tablets, ointments, and cosmetic preparations. In the chemical industry, which uses the remaining 10%, is employed as a foaming agent for the softening and treatment of textiles. Next is surface fermentation. Using rice bran or other starch or sugar rich substrate have been used for surface fermentation by Aspergillus niger or in liquid solution in flat aluminium or stainless steel pans. A niger can be used to produce citric acid despite the high content of trace metals in rice bran are used. The citric acid is extracted from the bran by leaching and is then precipitated from the resulting solution as calcium citrate. Next is submerged fermentation. It is a common practice where citric acid is made. The fermenter is made of acid resistance material which is stainless steel. The carbohydrate sources are molasses decatenized by ion exchange, sucrose or glucose, MgSO4, 7H2O and KH2PO4 at about 1% and 0.05 to 2% respectively are added. The pH is never allowed higher than 3.5.